Namaskar, I am Dr. Arumugam, Assistant Professor and Head, PG and Research Department of Botany, JJ College of Arts and Science, Autonomous, Pudukottai, Tamil Nadu, India. In this video, we are going to see about the somatic hybridization very detailedly. Somatic hybridization is nothing but development of hybrid plants through the fusion of somatic protoplast of two different varieties or species, which is called as somatic hybridization. The A plant is having the desired gene as well as B plant is having the desired genes. For an example, A plant is having the good variety, the B plant is having the stress tolerant gene. So when you are going to hybrid the A and B plant, we will have the AB plant. So the AB plant will be a high yielding stress resistant plant. So in this aspect we are going to have a new technology or method is called somatic hybridization. In the somatic hybridization we should isolate the protoplast. Before going to see the techniques of isolation of the protoplast, we have to know about the what is protoplast. The protoplasts are the cells which are cell wall removed usually by the digestion or the enzymatic method or mechanical method. In enzymatic methods, the cellulose and pectinase will be employed for digesting the cellulose as well as pectin. Because the pectin is holding the cell with one and another cell, so we should digest the or we should separate the individual cell, then only we can have the intact cell. From the intact cell we have to digest the cell wall. For digestion of the cell wall we used to use the enzyme is called cellulase. Once the cell wall is removed, we can have the protoplast in a spherical shape, what I have shown in this video. The applications of the protoplast is concerned, we can have the hybrid production. The hybrid is nothing but hybridization of two cytoplasms. For somatic hybridization, overcome the sexual incompatibility of the species or the varieties. In digestion, that is the foreign material into the cytoplasm will be incorporated. That is, we are going to ingestion of new foreign DNA or a foreign cytoplasm, which is consist of DNA, cytoplasmic organelle, whatever it is. We can have the add new foreign material. For DNA transformation, we used to study the wall synthesis or decomposition. But here, it will take part. The studies of the single cell system, we have to study over very well. This is a protocol we have to have for performing the somatic hybridization technique. The first, almost the isolation of the protoplast. As I said earlier, the isolation of the protoplast will be undertaken by two processes. One is enzymatic and another one is mechanical method. Second one is the fusion of two isolated protoplasts of desired species or desired varieties of two different characters. Identification and selection of somatic hybrid cells. Then we have to culture the hybrid cells, and we have to regenerate the hybrid plants or generator plants by using the somatic hybridization technique. This is a process for isolating the protoplast. What I mentioned earlier, one is mechanical as well as enzymatic. If you perform the both experiments or the both steps or both types of isolation of the protoplast, we can get the spherical protoplast intact. By mechanical method, we used to take the plant tissues, we have to do the cell plasmolysis, then we have to do the microscopic observation of the individual plasmolyzed cells, and we have to cut the ends or cut the cell wall of the isolated or plasmolyzed cells by using the knife. If you get the release of the protoplasm, we have to check whether our isolated or trapped or taken protoplasm will be intact without any disturbance or without any breakage. We have to assure that. And finally, we have to collect the protoplasm. This is the illustration of the mechanical method of plasmid isolation. First, we can see the cells were broken into new intact plasmolyzed cells. So, we used for the vacuolated cells like onion bulb, radish, and beetroot tissues. The mechanical method of protoplasm isolation used to yield the low yield of protoplasm. It depends upon the laborious as well as tedious process, because we have to cut the cell wall with a knife by seeing on microscope. So if the skilled person may be getting the high yield or may be getting the intact protoplast. By mechanical method of protoplasm isolation is concerned the low protoplast viability. In the case of enzymatic method of isolation of protoplasm, here the leaf will be sterilized and we should remove the epidermis. We have to do the plasmolyzed cells. We have to perform the pectinase as well as cellulase. The protoplasm released is isolated protoplasm as in the form. Again, in another type, first we have to treat with the pectinase, then we can get the release of the isolated cells. So, an isolated intact cells with the cell wall will be treated with the cellulase. Here we can get the protoplasm. So, first method we can add both enzymes in a single step. In alternative method, we can add the pectinase and then we can remove it and we can add cellulase as a second step. Anyway, we can get the high amount intact active protoplasm in this method. This is a process we used to follow in enzymatic method, which is used to the variety of tissues as organs including leaves or petiole or leaves, fruits, roots, coleoptile, hypocotyl, stem, shoot apex, embryo, or microspores. Among that, the mesophyll tissues are the most suitable or the best explant or source of plant material for isolation of the protoplast. In here we can get the high yield of protoplasm. It can be performed very easily. Then finally we can get the most of the protoplast will be viable. In enzymes for breaking of cell wall, the various enzymes will be employed depends upon the organisms. In the case of plant cell, we used to use cellulase, pectinase or mesozymes. In the case of bacterial cell wall degradation, we used to have the lysozymes. In the case of fungal, it would be degraded by the novozymes or glucosinase or chitinase. The streptomyces is actinomyces cell wall degraded by the lysozymes as well as achromatopeptidase. Okay sir, I have taken the protoplast as in the intact manner by using the enzymatic or mechanical method of protoplasm isolation. Followed by this method, we should purify the protoplasm by removing the debris like undigested materials, also burst protoplasms, because certain protoplast will be broken, like the broken protoplast will be removed on excess of enzymes or enzymes. The debris are removed by the filtering the preparation through the nylon mesh. Enzymes are removed by centrifugation, whereby the protoplast settle to the bottom of the tube and the supernatant removed with the help of pipette. Then it will be pipette out. Intact protoplast are separated by the broken up protoplast through centrifugation and removed by the pipettes as they are collected as in the top of the tube. And after purification process, the protoplast fusion will be undertaken. The fusion of the protoplast of the two different genomes may be by two processes, by spontaneous as well as induced fusion. The spontaneous fusion is a type of protoplasm fusion. It will be happen or it will be induced in between the intraspecific or the intergeneric. But in the case of induced fusion of protoplasmic fusion, we used to do that three. One is hemofusion and mechanical fusion and electrofusion. So the induced fusion will be undertaken any or what type of the protoplasm was isolated. So there is no need to be a intraspecific or intergeneric. Whatever the plant variety, we can fuse together. The methods of protoplasmic fusion. The protoplasmic fusion classified into two categories. As I said earlier, one is spontaneous. It is often fused spontaneously, fused through their plasmodesmata. And second one is induced fusion. The fusion with the help of the fusion inducing the chemical agents. Normally do not fuse with each and another. On hemofusion, in several chemical it has been used to induce the protoplasmic fusion, such as the sodium nitrates, and polyethylene glycol, calcium ions. These cause the isolated protoplast to adhere each another. It is a non-specific, and also it is an inexpensive. It can cause the massive fusion products because we can have a massive fusion products. It can be cytotoxic and non-selective, and also it having the less fusion frequency. What about the electrofusion? Because here electric charges or minus or plus ions will be used. Here the electronic stimulation will be used. The strength of the electric field will be 10 kVm, 10 kV per m. The electrofusion is very easy to control because when we can add the electric power range, so it can be controlled. It gives the reproducibility. It has the less toxicity because of we don't have use any chemicals, so the chemical influence will be very less in the case of electrofusion. The equipment is sophisticated and somewhat expensive. This illustration is coming to say about the mechanism of the protoplasmic fusion. First, having the two isolated harvested cells, the count has to be adjusted in numbers. We have to mix the cell and it has to be transferred into the fusion chamber. We should use to give some alignment, pulse, and also we have a post alignments. And finally we can have the transfer of the cell culture petri plates or tissues or any vessels. Okay sir, what about the fusion of protoplasm isolated from the bacterial cell? In bacterial protoplasm it can be obtained. Fusion can be carried out with a low frequency in some gram-positive organisms. In the case of gram-negative organisms, it is possible to obtain the protoplasm, but in regeneration is very difficult. Since it is a prokaryotic organism, the regeneration capacity is somewhat lesser than the eukaryotic organisms. The procedure is highly efficient as well as in high yield up to the 80% of transformants. The culture of protoplast. The culture of protoplast first we have to culture the isolated and transformed fusion undertaken cells into the culture vials. Such clumps, they are to the roots generate the complete plant depends upon the species. The plants are regenerated through the organogenesis from the callus. The microcalyx can be made to develop the somatic embryos which are used or which is converted into the whole plant through the germination. Okay, sir, good. We got the technology or method or protocol of the somatic hybridization. What would be the advantages? The advantages of the somatic hybridization is concerned the production of novel interspecific or intergeneric hybrid. For an example, tomato. The pomato word is coming from the potato as well as tomato. So we can come to know that the potato protoplasm is isolated from the good variety of potato. A protoplasm is isolated from the tomato. So both protoplasm will be fused together. We can have the pomato. The production of fertile diploid and polyploid from the sexually sterile haplines, triploids as well as aneuploids. The transfer of genes for disease resistant, abiotic stress resistant, herbicide resistant and many other quality characters may be incorporated. The production of heterozygous lines in the single species which cannot be propagated by vegetative means of propagation. The studies on the fact of the plasma genes. The production of unique or same hybrids of nucleus or cytoplasms. Good. What would be the limitations of the somatic hybridization? Means by the results it is a poor regeneration of the hybrid plants would be taken, would be derived. Non-viability also in some cases and some times we can get the non-viable of fused products. It is not successful in all the plants. Since we have succeeded both tomato as well as potato, but if you want to do that, brinjal as well as pumpkin, still it is difficult to produce new products, new variety or new species. The production of the unfavorable hybrid. So in some cases we used to have the unfavorable hybridization process also happened. The lack of efficient method for the selection of hybrids. No confirmation of the expression of the particular traits in some hybrids. So these are the some of the limitations or demerits or disadvantages of the somatic hybridization. Okay, good. What are the factors which influence the protoplasmic culture? Means first one is plant species and variety, and second one is the age of the plant as well as the part or organ of the plant. The age of the donor plant or the organ of the plant is a very big matter, because the availability of the plant material or the age of the plant, if you go and do that hybridization process, it may not be success. The pre-culture conditions, the protoplasm culture are highly influenced by the climatic factors as well as the different cultures of seedlings yields protoplasm having the different responses when it is cultured. And also fourth one is pre-treatment to the tissues before isolating the protoplasm. Because we used to use or we used to treat the cold treatment, plasmolysis and hormone increases the changes of the recovery of the viable products and their plating efficiency. And fifth one is density. It is a very crucial as it is influenced the plating efficiency and survival of the protoplast, and higher density protoplast complete with one another, while at lower density losses of the metabolism of the protoplast which is more. Happy? Somewhat we have completed the methodology or protocol of isolation and fusion and culture and regeneration of the protoplasm, that is somatic embryogenesis. Now we move into the new topic. I don't know whether I can cover all those things, but I can touch with the plant biotic. It is a very interesting plant bodies. Those days we used to depend the antibodies which is from the animals or bacteria, whatever the other than the plant material. But here, by the advancement of the biotechnology as well as plant tissue culture, we used to have the plant bodies, that is plant bodies. Plant body is nothing but it is antibody which is produced by genetically modified plants. They are a part of animal immune system and produced in the plant transformation, both of them with antibody genes from the animals. It was first discovered or first undertaken in the year 1989 with a mouse antibody made by a tobacco plant. Although the plant do not naturally make antibody, plant antibody have been shown to function in the same way as the normal antibodies. The plant are being used antibody factors using their endomembrane as well as secretory system to produce a large amount of clinically viable as well as clinically visible proteins. The antibody can be expressed in the plant as either length of the molecule or in a full form or in a very smallest fragment. As the full size monoclonal antibodies recently produced in a transgenic plants from the tobacco plant IgG against the nematode plant pathogen resistance, the purpose is concerned. The tobacco is Streptomyces mutans, it is SLGC, it's a therapeutic purpose. The plant sample is soybean as well as rice, IgG, that is herbs as simplex virus is for the therapeutic purpose. Tobacco, IgG, this is colon cancer, the therapeutic purposes. Alpha, it is IgG, is a human IgG for a diagnostic. So we used to diagnostic the certain protein. For that we used to apply it. The plant tobacco IgG for the rabies virus, the therapeutic purpose, a systematic injection may be made. The tobacco IgG is a hepatitis B virus. This is also for the purpose of therapeutic, and tobacco IgG is a low molecular weight phosphonate ester. It is a catalytic antibiotics. Okay, sir, good. What would be the method for production of plant body? The using transformation and the transgenic expression to induce the new genes into the host cell. This is a diagrammatic illustration. We can have the plant and we have to isolate the protoplast. Then we have to do that particle by the gold or the tungsten. And we have to perform the DNA isolation. A particular by using the particular current, we have to induce the that is desired DNA fragment into the isolated protoplasm. So there will be a transformation. The transformation plant will be cultured, a tissue culture or in the culture culture. So we can have this some new plant after the process of acclimatization. So we have to purify the plant bodies. Okay, sir, we made the transformation. After the transformation, we should isolate or purify the antibodies which we were incorporated in the transformation process. The purification can be purified cheaply in the large amount. Transgenic seeds assure that excellent storage properties and due to the limited range of the endogenous protein in the seed preparation as well as separation of plant bodies less complicated. There is no risk about the stated as animal disease to human as the antibody produced by the plants, because since it is the nature of plant association, so it is not be a toxic to the human. The techniques for the purification of the plant body by process is filtration, then immunofluorescence by checking the immunofluorescence by using the chromatography, and also defiltration and also polymer fusion. The methods will be undertaken by the process what we have shown in this video, and we should evaluate the technique transformation techniques has happened by using the technique RIA, ELISA as immunofluorescent southern plotting. We can perform the evaluation process. The production of cost of the plant bodies. Production of cost, the hybridomas, $1,000 per gram. Transgenic animals, it will cost of $100 per gram. But using the transgenic plants, we can produce the plant bodies by $10 per gram. So we can calculate the cost effective of plant bodies. Advantages: the large scale production plant bodies is immunotherapy, beneficial to the human, plants as well as the economical aspects. There is no risk of transmitting the disease to human. Then we can have the eliminated the toxic compounds. Applications: the application could be the antibodies generated by the plants are very cheaper, very easier to manage and safer to use than those that have obtained the animals. The treatment of illness such as the immune disorders, cancer as well as inflammatory disease also very easy. The plant bodies are very close to passing the clinical trials. The tobacco derived plant bodies entered to fight against the Ebola virus. Yes, it is ready. It is in practice now. Thank you so much.